I'm assuming you can see and hear me. Hank, can you give me a thumb up? Super. Let me share my screen. Great. Okay. All right. So I hope you can hear me loud enough. It's great to be here, at least by Zoom. I'm really sad that I'm missing you there in person, and which would have been much more fun and much more productive to talk to you in person. I want to use the next 30 minutes to just sort of share some thoughts on genetics in general and in spastic paraplegia. So I hope you find this helpful. And let me start. So, obviously, the big goal here is that we get to therapies eventually. So you may ask yourself, so what is the role of genetics in the current times of spastic paraplegia? What role does it play? What role can it play in the future? Oops. Okay. And so here are sort of three main topics that are pretty obvious. One is, of course, diagnosis. This disease is an inherited disorder, so genes play a key role. And as you know, there have been many genes being found, and they have a major impact on families and patients who actually do know their diagnosis or don't know the diagnosis yet. So when we have a gene identified, this also means immediately there is a target, what we call a target, meaning there is a specific molecular mechanism or place where we can think very specifically, scientists, pharmacological companies can think about how to influence this target. So it's very important for any rational therapy development that we know the genes and the genes in each person. And the third point is certainly the biology. Much, if not most, of what we sort of know about the biology of HSP has started with identification of these disease genes. Because when we know the genes, then we know they must be important. And then other scientists can really go deep into this. And these are often studies that take a long time. But we learn about the biology, and knowing the biology, sort of the mechanics of the disease, gives further opportunities, of course, for therapy development. So, and at the bottom of the slide, sort of, we need to know the exact diagnosis, which is the gene, the HSP gene that you may have. But there are also other layers, what we call modifier genes. So there could be secondary genes in a person that determine if the onset is earlier or later, or if the disease, for instance, is more severe. So, and in all of this, ancestry can play a role. And of course, clinicians and also patients are very interested in this relationship between the actual mutations and the resulting phenotype. And this is a developing story. We're not even close to finishing this. And this hopefully will then lead especially to genetic therapy solutions. So the other thing to consider is we are here at the Spastic Paraplegia Foundation meeting, and we talk about spastic paraplegia or HSP. But that is not a disease in a silo. In fact, it is sort of embedded, if you will, in a spectrum of similar disorders that either look similar or they affect similar structures in the brain, or they are caused by the same genes sometimes, and they can sometimes the same gene can cause HSP or one of these similar disorders. So here's sort of one sort of agreed on spectrum that is out there, and you can see that HSP falls right next to things like DHMN. What is this? This is distal hereditary motor neuropathy. or on the right side, there is ataxias, especially cerebellar ataxias. So each of these genes, they have their own spectrum of genes that they cause, but we are more and more aware of the overlap, and that HSP is not an island. It's connected with similar diseases. So benefit is, of course, we can learn from other disease fields. We can learn from them. We can learn from the biology that they identify. We can learn about the genetic targets and so on. So as most of you are well aware, HSP or SPG has turned out a disease which is not one disease, but many. And they are pretty much defined by the major underlying gene. So you can see here, this is not even up to date, that there are well over 50, the last number here is over 90 different SPG genes are known today. And some of these genes are quite related, so therapies might actually target a couple genes at once. But mostly right now, we probably would admit that each gene might need its own strategy for therapy, which is a daunting task, of course. Now, on top of this, unfortunately, many SPG patients don't know the genetic diagnosis yet. In fact, about 30 to 40%, it might even be a higher number of patients have not received a genetic diagnosis, even though they underwent the latest genetic testing available. So that is certainly something where we want to contribute more in the future. So genetics, is a lot in the media for many years now. Partially because it's so fascinating, of course, it's so impactful. Fortunately, there's a lot of attraction also in terms of technology development, and that has led to better and better technology to what we call sequence the human genome, a single human genome. Sequencing basically means measuring every single base and it's where it is and what it is. And that's the way how we find these mutations in HSP genes. And that's also the way how we find new HSP genes. And every year, there's a couple new HSP genes that are being discovered. So very important, the cost here, these are dollars, and this is what we call a logarithmic scale, the costs have declined immensely. So that today you can sequence what we call a regular genome for $500 or less. Here are some of the big company names. And you might be surprised, only a couple of years ago, only 2 years ago, the human genome was finally completely sequenced. There were always bits and pieces, they were still not sequenced fully. And so in this paper here on the right side, they describe the final blueprint of the human genome. So why did it take so long? Because the technology available, which we call short-read whole genome sequencing, is fantastic, but it had turned out it cannot sequence the entire genome. So we had to wait for another technology called long-read whole genome sequencing, which is now only the last year to mature enough to be used more widely in research studies. And I expect this also to enter the clinical testing realm. So here's an example how discovery in genetics has been developing over the last 20, 30 years. And as you can see in these different colored bubbles here, it's not a linear situation necessarily. And each bubble represents a prevailing technological approach in that time. So in the 90s and early 2000s, there was this so-called linkage, golden age of linkage analysis that were mainly family studies. And then it actually slowed down, arguably, it might have slowed down somewhat in the last few years. And there is a new technology now, the long-read genome sequencing, that is already showing much benefit. So another way to think about it is in this graph here on the left side, there is what we call the diagnostic gap, right? This is the clinical genome panels that are available that many of you have had. And if they're not successful, there is this gap. And then you may enroll in a research study, in a genetic research study, and if you're lucky, we find more and always find more, but it tends to be fairly small additions, any new gene. So the question really is in the genetics field, so where are the genes that fill this gap, right? And it has turned out that there are elements in the genome that we are not good at studying. And this has also to do with my first slide, or with an earlier slide where I showed you the whole genome wasn't even finished until 2 years ago. So we didn't have an opportunity even to look there. And here's another way to think about this. What clinical tests do and what most of we do in research today is, frankly speaking, only looking at about 2% of the genome. When we do whole genome sequencing, like some of you have undergone, of course, we look at the remaining 98% as well. But it has turned out we just, maybe the most important pieces of these 98% we weren't good at looking at. So again, coming back to the long read sequencing, and especially what we call the repetitive regions, they make up about half of the entire genomes, and only now we start beginning to see more of what is going on there. So, so what do I mean by all of this, and what does long read sequencing mean anyway? So the human genome, as you know, consists of these 4 bases that are arranged in a specific pattern. But it turns out there are regions in the genome that are, we call repetitive. For instance, there could be a CAG. Imagine 3 letters, CAG, and then CAG, CAG. So there's a long stretch of CAGs. And when we measure this, the machines we use to get confused, right? And if this is a very long CAG stretch, the read maps we what we call align to the genome, these short reads, we don't quite know where to align them correctly on these repetitive regions. And that has been, simply speaking, the problem. So certain genes like this FGF14 gene, you could simply not read out with current technology. There are some with certain tricks we can apply. There are certain things we can read, these repeat elements, like in Huntington disease, but not in all of them. And straightforward, really, these long read sequence technologies, they don't produce 150 letter or base pair long reads. They can produce reads that are over 10,000 base pairs. And suddenly you can really align them quite well and with certainty across these repetitive regions. So now we can read all these genes. So that's sort of breaking it down somewhat. And there's some more detail here on what kind of technologies are there. I'm not probably going to skip this. It's very technical, fascinating, however. And what we also observe is in the last 5 years, even though the interest in the 98% in the non-coding genome is high, has always been high, but mainly through some very sophisticated software approaches and improvements, there have been some really nice discoveries. For instance, on the left side is a gene called RFC1, which is a non-coding change. In the middle is a gene I think we talked about last year, possibly. It's a gene called CMT for CMT, and on the right side is a gene for ataxia. So patients with these genes, they can have spasticity, but it's not really the main idea here. I'm showing you this because all 3 of them are either non-coding, or the one in the middle is something where there is a gene that's almost identical. It's like a mirror gene that confuses us. And these gene pseudogenes, we will call them also, they are another source of confusion in genetics that we probably gonna solve much more in the coming years. So again, the main point really here is these three examples, they turned out to be not another rare cause of these disorders, these rare diseases. They turned out to explain a lot of patients, like thousands of patients worldwide. And so my point is, there is good hope that the 30 or 40% of patients who don't have a diagnosis today, it might sometimes go faster, so that we might reach much better diagnostic rates faster when we study these non-coding regions. So as you can imagine, as geneticists, we try to be at the cutting edge. So we work pretty heavily in this area right now. And we've been involved in a pilot project with the National Institutes of Health, where we study a few thousand of these long-read genomes and see what's in there in general. And we see, let's say, signatures that we haven't been able to see before. And it's a working group, not just the Miami group. And everybody's really quite excited and surprised what else is out there in the human genome. It's not like we know the human genomes that well yet, it seems. My student, Sarah Fasal, she just graduated last year, but she developed an AI, an artificial intelligence-driven program to go faster through these new findings. So these things also happen, that we're trying to use these machine learning approaches. And then here's another, hopefully not too confusing aspect of these sort of new genetics. Some of it is something geneticists have known for a long time, but now we can study it in much greater detail. So this is also not spastic paraplegia, it's another gene recently identified. And we were able to sequence the parents and the affected child. And what we see is for this particular gene here, which is a non-coding change, is one of these repeat changes. When you inherit this chromosome with let's say the mutation, what happens is this repeat always gets smaller. It always shrinks. We don't quite know why, but that is the case. When you get it from your father, it shrinks. When you inherit it from your mother, it always grows, it expands. And when it grows, it's not good, because when it grows over a certain size, these repeats, then they cause disease. So, but when you imagine this, that you study a genetic disease where every generation changes, and so, depending on where you get it, from your dad or from your mom, the pedigrees and the family history becomes quite confusing for everybody involved. And that makes it, of course, complicated, especially if you don't know what you're looking for. So this is the kind of genetics we have to really think harder about going forward. And this is another detail out of these pilot studies on these long-read genome collections. And these are all from, or mostly from controls at this point, actually. They're probably less than a few thousand long-read genomes produced from patients worldwide so far, at least at high quality. So this is all fairly new. So when we look at these repeat loci, which we were not really able to sequence so well up until now, everybody who sees these data is amazed, because it's not like CAG, CAG, CAG. When you look closer and with great detail, you see these motifs are interrupted. So each color is sort of a different motif, and you see down here the motifs. And it can get quite complicated. They are stacked into each other. So each line here, each row, is a different chromosome. So there are many patients stacked up here, or many individuals, and you can see everyone has a somewhat different type of of repeat at this place. It's conceivable that one of these actually is linked to disease. But finding these, you can imagine, is even harder maybe than what we have done so far in genetics. But now at least we are able to see that, and we can start really thinking about it. Another very important topic is, of course, you may get back a genetic report, and with 90 HSP genes currently known, so the test may have tested 90 different genes at once. Chances are that there are changes, I'm not saying mutations, just changes, that where the clinical laboratory cannot be sure, is this a benign variant that is there by chance, or is this actually the one pathogenic, the one mutation we really look for here? And when the clinical lab is not sure what to do with that, they call it a variant of uncertain significance, or VUS, or VUS. And this has become quite a problem. The correct classification of variants in known HSP genes can be quite tricky. And there are many, many patients out there who don't quite know what their gene is. They have a VUS in their report, and their doctor doesn't know either. So how can we better classify, basically sort into the benign bin or in the pathogenic bin these many variants? That's a task, and that is turned out not to be an easy task. So this is another thing we work on, and the field is working on and thinking about a lot. There are traditional ways, like this is a schema that clinical labs use. It's quite detailed and can be confusing, how to classify variants. There are newer approaches or alternative approaches that are being tried out. They're not really fully accepted yet, I would say. And many have to do with machine learning or artificial intelligence, a bit like ChatGPT, frankly. And the judges are still out there if that will contribute to a better diagnosis, a better classification. I think there will be some sort of convergence of all these different approaches in the future. Here in Miami, we have published one of such AI or neural network approaches to classify variation, and we were surprised how well it actually can work. So I don't want to go into detail. This is a published work, and we use it on a regular basis. In fact, it helps us to identify novel genes. There are about a handful of genes that where the computer basically told us, you should look at this gene. And then we look at this gene, and in a handful of cases, it turned out to be, yes, that was a good idea to look at this gene. This is, in fact, a novel gene. So another thing you can do when you start trusting your machine learning more, you can actually go out to a gene like spastin or SPG4, and you can say every conceivable missense change in spastin, predict right now, is this pathogenic or is it not pathogenic? Is it benign? And we did this for spastin, and this is what you get. So this is the gene here, and each dot is a different missense change that is theoretically possible. And all the red dots are the ones that are known mutations, missense mutations in spastin. And where these known changes, when they were considered disease-causing, they end up high here and low here and also low here. And you can see that the red dots, they correlate pretty well with the gray dots. So if we start trusting, oh, this works pretty well for the known spastin genes, then maybe the other gray ones are not so bad either. So that's really where I'm going here. And finally, I'm sure Darius has told you already about our new consortium, the SP-CERN. And the basic idea really is to bring people together, experts, make them work together to create common resources like clinical databases, DNA biobank samples that are shared in the group, but also genetic data. And so for genetic data, bringing genetic data together, that makes it easier and more successful to combine them and to find important things in these data. So we're going to use the Genesis database, which in Miami we have used for over 10 years now with great success. And it already contains a lot of data where the SP-CERN can sort of add on to that. In fact, right now there are over 2,000 genome data sets in this Genesis database. And they're not in some hidden hard drive. They actually de-identified in a cloud, and they highly protected, but we can share them easily, and we do share them easily among scientists in SP-CERN, but also beyond with colleagues in other countries who are researchers. So only researchers have access to these data. And this is one example how this has worked already in the past. This UBAP1 gene is a HSP gene, and it was found through really collaboration with different countries because they found each other very much so on this Genesis database. So it's very powerful. And finally, a word on other alternative approaches. Once we have a lot of genetic data, we can also start using statistics. We know from other diseases who have been successful in creating large genetic data sets that you can use statistics to find signals for disease genes. And this is sort of a pilot study we have performed a couple of years ago, which also included HSP. And you clearly start seeing sort of differences based on statistical measures. It's still a jump from there going to individual genes, but it is a very worthwhile approach. I think it's just hard in rare disease because you need to really build up a large genetic database. And another topic that every year comes up and we are very aware is the fact that, for instance, in spastin, which is a more common HSP gene, even patients have realized that there are large differences from patient to patient. And doctors have always asked, why is that? What explains this difference? Even in the same family, which carries the same mutation, you see differences, later onset, earlier onset, and so on. And the idea is there could, of course, absolutely be environmental modifiers. And environment really means everything from infectious diseases to food to climate, you name it. But there could also be secondary genetic reasons, so-called modifier genes. And the group in Paris has, and collaborators, they have performed the first SPG for genetic modifier study and published this. And in fact, they did find a signal here also, again, with these statistical methods. They found a signal in this gene called SARS2 that may contain genetic elements that modify how SPG4 is expressed in people. But I'm convinced this is not the only gene. This may not even be a main gene, but it's a great first find and needs to be followed up on. So we are in the SP-CERN. There's definitely interest to do that, and there will be resources to do that. A group in Germany, Rebecca Schüler with Treat HSP, she's working on this, and she's collaborating with us in Miami on that topic as well. So with this, I want to stop, and not sure how we're going to do it, Norma. I would be very happy to answer any questions if there are any.
Hey, Harper, can you type in the chat box for him to put up his email for people to send questions to him? Can you do that for me?
I can't hear.
You can open your mic if you want to talk to him.
Works.
Harper, you can probably speak to
I
him
will send you an
directly.
email. This.
Okay, real quick, can you capture, before you flip over, Harper, can you guys see that down there, his email? Let me read that out loud for you guys. All right. SZUCHNER at med.miami.edu. Okay, guys, if you have any questions for Dr. Zuckner, you can email him at that address. Okay? So he is going to unzoom, and we are going to move on to Dr. Calamé, who's next in the bullpen. So everybody hang on while we make the switch, and he knows that you guys are all clapping loudly for him because he did such a great presentation. So thanks, Dr. Zuckner. Okay, coming up, and there he is, Dr. Calamé. Here we go.
Can you all hear me? Should I get started? That a yes? Okay. All right. All right. Well, hi, everyone. It's really a pleasure to be able to speak to you all today. My name is Daniel Callum. I'm a pediatric neurologist at Baylor College of Medicine and Texas Children's Hospital. And today I'll be talking about my work on the genetic mechanisms underlying undiagnosed pediatric HSP. I'm sorry I couldn't be there with you in person today. I'd love to be there in Tampa. But I'm coming to you today from Houston, Texas. I work at Baylor College of Medicine, and I'm an attending physician at Texas Children's Hospital, which you can see here. TCH is the largest children's hospital in the United States. It sits within the Texas Medical Center, the largest medical center in the world. TCH is consistently one of the top children's hospitals in the United States, and our neurology group in particular is consistently one of the top 3, top 4 programs. I focus on neurogenetics. In particular, I focus on movement disorders and hereditary spastic paraplegia. And I'm very pleased to be a part of the SP-CERN natural history study. I work with Jim Lupski. I suspect many of you in the room are familiar with Jim. Jim is kind of a big figure in human genetics. He really created the field of genomic disorders with his discovery of the PMP22 duplication as the cause of Charcot-Marie-Tooth type 1A, the most common type of Charcot-Marie-Tooth or hereditary neuropathy. And you know that really really changed everything, and of course has had a significant relevance to HSP, since deletions and duplications of SPAST are one of the most common causes of HSP. And the SPF previously supported Dr. Lupski some years ago, and they published great work with Phil Boone, showing that there's some unique features of the genomic architecture around SPAST that predispose it to have frequent deletions and duplications, these repetitive elements in the genome called ALUs that help mediate recombination. And so Jim thinks very, very fondly of the SPF. And so our focus is really on undiagnosed patients. And in HSPs, like Dr. Zuckner mentioned earlier, we see a similar story to many other genetic conditions. There are many HSP genes. I'm aware of at least 83. Dr. Zuckner mentioned 90. But despite this large number, the reality is that with our best clinical genetic testing, we still don't get a diagnosis in many patients, if not most. The figure on the right comes from a group in France. They applied a gene panel approach to over 800 patients of HSP, and they were only able to get a diagnosis there in about 30%. When you look at the breakdown of the genes that are involved, there's just a couple of genes that really make up the lion's share of diagnoses. Of course, SPAS being the biggest one for dominant HSP, and then for recessive SPG7. And the reason for that is for SPAS, of course, again, there's features of the genomic architecture that predispose to deletions and duplications of it. For SPG7, there's just simply certain pathogenic variants that are more common in European populations that make it to be one of the most common recessive ones. But when you get outside of those couple of genes, you have many ultra rare causes of hereditary spastic paraplegia, and it's clear that there are many more that remain to be identified. So as we think about how to get answers for undiagnosed patients, it really comes down to two areas. I think Dr. Zuckner talked already a lot about this first area, the idea that we're missing certain genetic variants, things in deep in the introns, in the non-coding parts of the genome, the structural variants, the repeat expansions. And we believe that new technologies like whole genome sequencing, especially long-read whole genome sequencing, RNA sequencing, and refinement of our ability to analyze the data, it's going to make some significant breakthroughs in this area. But it turns out that in many cases, the answer is actually already there. It has already been identified in the exome or genome, clinical exome or genome that was already done, and it just simply wasn't recognized or wasn't understood at the time. And it because of the three kind of phenomenon that I list here, the HSP mimics, we call phenotypic expansions, as well as novel disease genes. And to get to the bottom of these, we really just need to get into the data, to reanalyze it with a more critical eye and with additional analytical tools, data sharing that Dr. Zuckner mentioned, and then a lot of experimental biology. And so I'm going to give some examples that have come out of our work in recent years of these three things. This first one is an HSP mimic. This was a story that began when I was a resident in the Bluebird Circle Clinic at TCH. I saw a young woman, 17 years old, who had a history of developmental delay and progressive spastic paraplegia. She first walked around 2 years of age with a walker. Her first words came around 4 years of age. And when I saw her at 17 years old, she had a diagnosis of mild intellectual disability, but her spasticity and her gait problems had really progressed, and she could hardly stand or walk without considerable assistance. And so she came to me already with the clinical diagnosis of a complex, complicated form of HSP. This figure shows the typical pipeline where we go from a DNA sample to sequencing to the computational pipeline by which that sequencing data gets kind of put back together again to yield the sequence of the individual's DNA. And then from that, we pull out the variants that are then analyzed and interpreted. And ultimately, whatever is thought to be significant of that goes into the medical report that goes to the patient and their doctors. But in cases like this patient, she had clinical whole exome sequencing actually very early on in the course of when exome sequencing was available, and her report was negative. And so, at this point, we kind of have a few options available to us. We can go back and sequence them again using a more advanced technology, like perhaps now long read sequencing, or we can just get the existing data and reprocess it in the lab to take a closer look at it and again apply new analytical tools. And what happened in this case was our reanalysis led us to focus in on this missense variant in this gene ATP1A3. This particular missense variant had never been seen before in any other patients in the medical scientific literature. It had not been identified in any healthy individuals in population databases. It was predicted damaging. It was a very conserved nucleotide and amino acid. And it fell in a part of this gene that was kind of a hotspot for other pathogenic variants. So it seemed to be a good fit from a genetic perspective. But the problem was that it didn't seem to be a clinical fit, that it didn't fit the diseases that were currently linked to this gene at that time. And that's why it didn't make it onto the clinical report. For the neurologists in the room, especially the movement disorder neurologists, this gene is very well known to us. It's a common cause of very striking paroxysmal movement disorder syndromes. On the left, you see an example of the most common one, alternating hemiplegia of childhood. This is a patient that I see in my clinic, and I share these videos with the permission of the family. What happens in this condition is in the first year of life, children will have hemiplegia. It'll come on, weakness on one side of the body. It looks like a stroke. That's kind of what you can see in the video on the far left. If you look real carefully, she's having a hard time using the left side of her body. And that will last for minutes to hours or even days. And then it goes away, or it moves to the other side of the body. It's a very striking condition. There also are many other abnormal movements. I'm also highlighting abnormal movements of the eyes that come and go. But this is what we see with ATP1A3, these very striking episodic movement disorder phenotypes. ATP1A3 encodes a pump, a sodium-potassium pump that's found within our nervous system. Maybe you see a figure of it on the right side of the slide. And this is important for maintaining electrochemical gradients in the brain. And so, again, the issue here was that it just didn't seem like a good match with the patient's condition, which looked more like a slowly progressive hereditary spastic paraplegia. And so the way we approached this was to then analyze data from clinical diagnostic lab databases, as well as to reach out to research collaborators from around the world. And it turns out that this particular variant is a fairly, I'd say somewhat common recurrent variant that causes this sporadic complex HSP-like disease. We found it in a total of 9 families. In several cases, we were able to show that it was a de novo mutation, a new mutation in the patient. And when we look at the diseases in these patients, it really looks like complex HSP. They all have developmental problems. They all have spasticity. Most of them have spastic paraplegia. And then in the patients who have spastic paraplegia, it's pretty evenly split between a progressive form and a more static form. And really, we don't see very much in the way of these episodic paroxysmal movement disorders like in the other ATP1A3 related conditions. We worked with another pediatric neurologist and physician scientist, Sho Yano, at the University of Chicago. Sho is very interested in this gene and is a great electrophysiologist. He was able to show that this variant completely kills the pump activity of the gene, similar to other pathogenic variants in ATP1A3. But what was really fascinating was he also found that the variant does something that hasn't been seen before. It results in an inward leak of cations, of ions, across the plasma membrane. Again, this has not been seen before in any other ATP1A3 variants. And in theory, this should counteract the activity of the other normal copy of the gene, and it may explain how this variant causes disease. So to summarize the HSP mimics, I think it's important to recognize that other genetic disorders can be clinically almost indistinguishable from pediatric simple and complex HSP. And because of this, there's really a big need to use untargeted genomic sequencing like exome and genome sequencing for clinical diagnostics in pediatric HSPs. I'll next move on to an example of a phenotypic expansion. This one focuses in on a gene called FLVCR1. FLVCR1 is a transporter. It's found in most of the cells in our body, most tissues, and it acts to transport 2 metabolites, choline and ethanolamine, into our cells. Choline and ethanolamine are critical for many processes. In particular, they are the substrate for what's called the Kennedy pathway, a pathway that generates phosphatidylcholine and phosphatidylethanolamine, two critical components of cell membranes. Choline is also the substrate precursor for acetylcholine, a neurotransmitter. And choline also plays a broad role in many other metabolic processes as well. So very, very important gene and protein. We've known for some time that this is essential for life, at least in the mouse. When you knock this gene out in mice, you don't find any mice that are knockouts, that don't have the gene at all. So if you look earlier during embryonic development, you can find some embryos that lack the gene. But as you can see here, they're very small. They have severe skeletal malformations, and they have very severe anemia. You can see that embryo is very pale compared to the normal one on the other side. In contrast, in humans, this gene was linked 2 years later to something very, very different, to a childhood or adult onset syndrome, where patients develop ataxia, progressive vision loss, blindness, and sensory neuropathies. It's a recessive disease, as shown in this pedigree on the left. And since this initial paper back in 2010, there's been at least in their 30 families identified, probably 40 or 50 patients with this condition, but very different from what we see in the mouse. And so we became interested in this because in the course of our work, both on HSP as well as in other severe neurological disorders, we kept finding patients who had really impressive variants in this gene, but with very different diseases. And so what I'm illustrating here is all the families where you see the red boxes or circles. These are families with very, very severe neurological disorders. I'll show you what I mean by that in a little bit, but you can see, unfortunately, that many of the children had passed away in the first years of life. We also found some other families that had the more typical disease, the ataxia and sensory neuropathy, but even in the kind of the milder side of the spectrum, we were finding some diseases that hadn't been associated with this gene yet. This patient in particular has HSP, has simple HSP, began in childhood, slowly progressive, none of the other features associated with this gene so far. And these two families have more of a classic kind of Charcot-Marie-Tooth motor predominant distal motor neuropathy. And so I'll now explain to you what I meant by the severe end of the spectrum. So these are some pictures of affected children as well as their brain MRIs. These children are very developmentally impaired. They almost all have very severe spasticity, very severe seizures. And when you look at their brain MRIs, you can see they all have evidence of brain atrophy. There's quite a bit of variability in the appearance of the brain MRIs, but on the most severe end, like you see in the bottom right, it can be very, very severe. Like this child just had a very thin rim of brain matter, kind of like what we call hydranencephaly in neurology. We also found the most severe disease, what we believe is the most severe disease from this gene, in two families. There were two families where there were children who were stillborn, who had significant skeletal malformations, anemia, and severe brain malformations as well. And in these families, they had mutations that caused them to have a complete lack of this gene, so analogous to what we see in the knockout mice. And we studied the impact of the missense variants that we had found, the mutations on the protein function and choline and ethanolamine transport. You're seeing on the far left that actually these missense variants don't seem to impact the stability of the protein or its localization to the plasma membrane, but nearly all of them significantly reduce choline and ethanolamine transport. And some of them, I don't show the data here, but they also impact the splicing of the gene, and therefore either result in a truncated protein or perhaps no protein at all. And so kind of in summary, we think that the severity of disease from this gene is a consequence of the severity of the mutations that the patient has, with more severe ones causing more severe disease and the milder phenotypes from the milder missense variants. And so I think the summary here is that these phenotypic expansions, where a gene that was linked to one disease is then found to cause other diseases, this is really it really seems to be the rule in genetics rather than the exception. Probably most genes are going to cause more than one disease, more than one inheritance model, both dominant and recessive. And so this is something we need to be on the lookout for in HSP, because I think HSP may be, in some cases, a rare manifestation of other genetic disorders like FLVCR1. And then finally, I'll wrap up with a gene discovery, and this is still a ongoing story. So I'm happy to share our preliminary data. This is about a kind of continuing on a similar theme of transporter disorders. This is a gene called SLC7A1. This is another transporter. It's found all throughout our body, especially in the endothelial cells that line our blood vessels. This one, though, transports a few amino acids, the cationic amino acids like arginine, lysine, and ornithine. It's a high affinity transporter. In this figure, I focus on arginine because there's a lot of important biology involving arginine that probably ties into this disease. Arginine is an amino acid, so of course it's also important in just building proteins, but it plays a particularly important role. It feeds into the urea cycle, which some defects in the urea cycle are known to cause spastic paraplegias. It also is a substrate for nitric oxide synthesis, nitric oxide being an important vasodilator, as well as the synthesis of creatine and polyamines. This gene, the mouse story is very similar to FLVCR1. When you knock this one out, mice die. They do survive till birth, but they die on the first day of life. And strikingly, they also have very, very severe anemia. This paper was published well over 20 years ago, back in '97. But really, since then, there hasn't been much work on the role of this gene in the nervous system. And so, in the course of our work of reanalyzing genomic data from undiagnosed patients of HSP, as well as doing genome and exome sequencing in new patients, we came across this, and we think this is a really strong candidate gene for recessive HSP. We've identified a total of 7 families so far. There's 11 affected individuals. Mostly they have homozygous missense variants. There is one compound heterozygote with a missense variant and a loss of function variant in trans. And the phenotypic spectrum, like in many of the HSP genes, is broad. It ranges from, in a couple of families, from simple HSP to another couple, more of a complex picture. And then there is one family, one patient, the one shown here on the bottom right, that has a very severe spastic neurodevelopmental disorder. I have a few pictures here just to show of some of the affected individuals. This child here is, cognitively, developmentally is pretty much where he should be, but just has progressive spastic paraplegia. His brother is similarly affected. They both have, on their brain imaging, they do have very prominent ventricles and some degree of Vermean atrophy. And then I mentioned this more severely affected child. She has microcephaly and pretty severe developmental problems and more severe brain appearance. When you look at these missense variants, we haven't seen any recurrent ones yet. We think this is a very rare cause of recessive HSP. But we do see, curiously, that there are recurrently affected amino acids, valine 291 and arginine 588 in particular. The missense variants all fall within transmembrane domains. They're all very well conserved across different organisms, as well as different genes within this gene family, the SLC7 family. They're all predicted to be very, very damaging. When you look at their distribution within the actual structure of the protein, this is from AlphaFold, and this also shows alpha missense predictions of how damaging each changes in each amino acid are, where the blue or white, blue is benign and red is predicted pathogenic. You see again that all the variants fall in areas that are predicted very, very damaging, very likely to be pathogenic, and they tend to be near the inner pore of this channel, which suggests that they're probably impacting the flow of arginine and other amino acids through the transporter. And so right now we've generated both constructs for all these missense variants, and we've also made knock-in cell lines, and we're doing arginine transport assays and other studies to understand the biological consequences of these variants. So in conclusion, I hope I showed that HSP mimics and phenotypic expansions are a real issue in undiagnosed HSP patients. And because of this, we need to be thinking about genome-wide sequencing over single gene and gene panels, especially in pediatric HSP. We and many other groups have shown that the reanalysis of existing exome and genome sequencing data can significantly boost the diagnostic yield and facilitate discoveries. And like Dr. Zuckner mentioned earlier, gene discovery in HSP remains brisk, one to three genes, I would say, just about every year. And in the process, we're discovering novel disease mechanisms and hopefully opportunities for new therapeutics. And so I want to conclude by acknowledging, of course, my mentor, Jim Lupsky, and all members of the lab. We have great leadership at Baylor Child Neurology, Gary Clark and Huda Zogby. We're a part of a large NIH rare disease consortium called the GREGOR program. If you have undiagnosed patients, or if you are an undiagnosed patient, feel free to reach out to us. We have ways to help get you research genomes, long read, short read, transcriptomics, et cetera. We have many collaborators I have to acknowledge, and especially the patients and families that participated in the studies. And then finally, I really want to thank the Spastic Paraplegia Foundation. Jim was awarded a grant from you all a couple years ago, and it was instrumental in this work and getting me involved in HSP. But thank you very much. And I will drop my email in the chat. Please feel free to reach out to me.
All righty, everybody.
All right. Thank you all.
Thank you.
Bye-bye.
Dr. Calamé, great job. I know that's going to go up there pretty quick, but Dr. Calamé's email is in your program under his bio, okay? So you can look in that if you guys want to reach out to him specifically, okay? And we do have Dr. Zuckner's email, but I have it in the back of the room. So anybody need that, come back and see us, and we can give you that, because that went by pretty quickly. Hey, I want to do a quick couple of things. Remember, everybody, 5:15, the 5K photo outside with Jim. Also, the PLS community, when they do their breakout session at 4 o'clock, inside of that time frame, they're also going to have their group photo done during that breakout session. Just wanted to remind you that. Hey, and something really cute and really great that I wanted to tell everybody about while we're making a transition to our next thing is it's Andrew Hendrickson's birthday today. Andrew, where are you? Stand up. There you go. Andrew has been doing a great job helping us throughout the conference, and he's been taking 1 million pictures, which will show up somewhere on our Facebook page and Instagram pages and our website, and next year's picture PowerPoint of our historical review of conferences. You'll see yourself in some of those in that too. So we're switching over right now to get ready for our next presentation, which is going to be, looking at the wrong day, Dr. Shearbaum, and we're working on that, getting her laptop hooked up right now, and then she's going to work her way this way, and then we're going to get her going on her program, okay? And then after that, we're going to have mini stretching with Zachary again, okay? So you guys are getting a lot of workout. And he, by the way, is also having his own breakout at 4 o'clock, between 4 and 5. So if you really want to go and get a nice workout with him, we're looking forward to that a little bit later. So once we get everything set here, we're switching between laptops and everything. Remember also, if you want some raffle tickets and t-shirts and bracelets, you have till 5:15. So make sure you stop by the registration table and get all that good stuff, okay? Because we have it and it's here and available for you. And then later this evening, during our dinner, we're going to have our raffle and our auctions and other kind of fun stuff. So that's going to get everybody really excited, and it's going to be a lot of fun. So it looks right now that I see we have the presentation up, and here we go. We're ready for Dr. Shearbaum. Here she comes.
Hello, everybody. My name is Luca Shearbaum. I'm a research fellow and pediatrician working with Dr. Darius at Boston Children's Hospital. And I'm here today to talk to you about one of our research programs, the HSP Sequencing Initiative. And with this program, we aim to close the diagnostic gap in childhood onset HSP. Some of the things I'm going to be talking about have already been mentioned by Dr. Züchner and by Dr. Kalami, but I'm hoping to show you some new perspectives. Next slide, please. So to give you an overview over my talk today, I will first talk about the evolving role of genetics in HSP, then briefly talk about HSP as a differential diagnosis in cerebral palsy. Then I will present you our HSP sequencing initiative, introduce you to our cohort, and I brought 2 of the cases we worked on this year. And then I will talk briefly about some future directions. Next slide. And next slide again. There is. Perfect. So with this slide, I would like to show you how the research on hereditary spastic paraplegia and on genetics has been evolving in parallel. So we see that the timeline starts on the left in 1880 and 1888 with the work from Stümpel and Lorraine, who first described and recognized the hereditary causes in spastic paraplegia. Stümpel described 2 brothers with hereditary spastic paraplegia, and Lorraine described the first 29 families with HSP. Then we continue with the description of the first HSP gene, SPG1. In the genomic research, we have the Human Genome Project in 2003, which sequenced the first human genome, almost completely, 92%, as Stefan noticed in his talk. In 2009, we established whole exome sequencing. And then in 2022, SPG91 was established, the SPARTAN1 gene to cause HSP. And so we see that over the course of 30 years, 90 disease genes for HSP have been discovered. And in the same year, the Telomere-to-Telomere Consortium managed to completely sequence the first human genome. And we see that the research has been accelerating towards the last few years. Pedro, next slide, please. Thank you. So with these 2 graphs, I want to show you how this acceleration in genomic research also leads us to new discoveries in HSP. So we see on the left side the cost of genome sequencing from Illumina, one of the big laboratories that researchers work with. So we see that 10 years ago, the cost for one genome was over $5,000, and right now it's approximately $200. per genome. And mirroring that on the right side, we see the PubMed research on HSP genetics. So 30 years ago, or 50 years ago, we had almost only like 5 papers that were published per year, and now we have over 300 publications per year on HSP genetics, which is amazing, I think. Next slide. To talk about hereditary spastic paraplegia as a differential diagnosis of CP, I brought you this meta-analysis from González Mantilla et al. They looked at 13 different studies that looked at the diagnostic yield of exome and genome sequencing in patients with cerebral palsy, and they found that indeed 30% of the patients that were diagnosed with cerebral palsy had a genetic diagnosis. But we also see that there's a broad range of the diagnostic yield between 10 and 40 to 60%. And it is important to notice that the diagnostic yield in a sub-cohort analysis is higher in pediatric patients with CP. And we also have to keep in mind that the most common genes where disease-causing variants were identified were the SPAS gene and ATL1. Next slide, please. So if we talk about the role of genomic or genetics and diagnosis, we often start with a really targeted approach, single gene sequencing or gene panel. And what we are often implementing now as the gold standard is the clinical exome sequencing. But the diagnostic yield of all these approaches right now is between 60 and 70%, meaning that 30 to 40% of the patients with clinically HSP don't have a diagnosis. Next slide. And this work of Cao et al. Shows us in more detail what this means. So in the patients that have a positive whole exome sequencing, most of the mutations are attributed to SPG4. If we look at CNVs, also most of the mutations are found in the SPAS gene, but 40% of the patient population that were looked at in this paper didn't have a genetic diagnosis. And the question is, what are we missing? There are several options. So one option is that we don't identify the mutations in genes that are already known to cause HSP. And the second option is that there are more genes out there to be discovered that cause HSP, or possibly as well a combination of those two things. Next slide. And to address this issue, to address the 40% of unsolved cases, we started the HSP Genomic Sequencing Initiative. In this initiative, we include all patients that are younger than 30 years old that have clinically suspected HSP but no genetic diagnosis. And how do we recruit out patients? So one contributor is the Movement Disorders Clinic at Boston Children's that is led by Dr. Darius. Then we have our natural history study. We have some patients and clinicians that contact us directly. And one big support for this are patient foundations that are really essential in connecting researchers and patients and their families. Next slide. Thank you. So what's the aim of this initiative? First, we want to establish the diagnostic yield in exome-level reanalysis in our well-characterized cohort with children and young adults with a clinical diagnosis of HSP and that don't have a genetic diagnosis. And the second aim, next slide, is to establish the diagnostic yield of short-read and or long-read whole genome sequencing in said cohort. Next slide. So this is an overview over the patients that we have enrolled so far. We have 95 patients enrolled. 68 of those are trios. 8 of those are trio plus, which means that we not only have parental DNA available, but also another family member enrolled. 10 singletons, 1 singleton plus, 5 duos, 2 duo plus. And important to note that 80% of the patients we have enrolled, we have samples from both parents available, which is really important to increase the quality of our analysis. Next slide. Thank you. And what I briefly mentioned before, we are implementing short-read whole genome sequencing. In short-read whole genome sequencing, the first step is a library preparation where the DNA is fragmented, adapter sequences are added, then we have a cluster generation by in situ amplification, and then we have in parallel sequencing by synthesis, which means that while the DNA is synthesized, we also get the sequencing data. And there are several strengths to this approach. So one is that it's an established approach. We have a low error rate from 0.1 to 0.5%. We have relatively low costs, and we can detect CNVs and non-coding variants. As a disadvantage, we know that we still have a diagnostic gap in detecting repeat expansions and complex structural variants. Next slide. And next slide. Right now we have DNA available for 51 trios, 7 trio plus, so we see that this mirrors the enrollment of the families in our cohort. Next slide. 70% of our patients have had previous genetic testing that was non-diagnostic. 13 of our patients did not have genetic testing, and from 10 patients, these informations are still pending. Next slide. So this is an overview over our analysis workflow. It's a little bit busy, but I'm going to walk you through this. So on the first up left corner, we have the data and sample collection. This means that we get the blood drawn from patients and parents. Then we take a short questionnaire to get a better phenotypical understanding of the symptoms of the patient. The phenotypical data will be stored on a REDCap project. The blood will be shipped to GeneDx, our collaboration partner. They will do the DNA extraction, and they will do the sequencing for us. Then the data will be stored on a Google Shared Drive, and then all the data will be moved to our research computing department. They do the quality control, they do the alignment and the variant calling with the Dragon pipeline. And those data, to analyze those, we have different tools. One tool is imaging that we use for a specific type of variants, for small nucleotide variants called SNVs, and for short indels. And then we have Genuity that we often work with. It's a tool for different types of variants, for structural variants and larger CNVs, and we also can do a cohort-level analysis. Then on the right side, you see the Genesis project. It's one of our collaborators, collaborations with Stefan Zuckner's group in Miami. They have built a platform for variant calling and analysis, and also for data sharing, as he previously mentioned. And we use all these tools in combination to increase the diagnostic yield as a complementary approach. Next slide. So, how do we approach if we see a variant that might be interesting? We start with strict filter criteria you can see in red, and then we slowly move forward to more permissive criteria you see in green. And we assess the variant starting with all red filter criteria for different qualities, for zygosity, allele frequency, combined check, and then also for associated disease. And in the end of this process, we tag the variant. If we decide this is not a variant of interest, we loosen our filter criteria step by step to more and more permissive. If at the end of all permissive filter criteria we still don't have any variant of interest, we will reanalyze this data set within the next year. If there is a variant of interest, we will follow up, which is mostly done through research collaborations. Next slide. And one critical part of this process is our monthly discussion as an interdisciplinary team with neuropediatric clinicians, genetic counselors, and clinical geneticists, and we discussed the genetic and phenotypic finding, always with the question, is this finding our diagnosis? And if we think and come to the conclusion, yes, this research finding might be the explanation for the patient's symptoms, we ask for clinical confirmation, called clear confirmation. If we think, yes, it's a possibility, but we're not quite sure yet, we determine which steps have to be done to confirm the diagnosis. This can mean submission to GeneMatcher, which allows us to collaborate with other researchers. We can do additional testing like muscle or skin biopsies. We ask for an ophthalmology consult, or we do additional imaging. And if we don't think this is a good genetic explanation, we will reanalyze the case in a year. Next slide. So this brings us to the first case that I brought for you today. It's about a 5-year-old girl that had a normal birth and prenatal history, but the parents noticed that she had some neurodevelopmental delay in motor, speech, and language. She had progressive lower limb spasticity with hyperreflexia, clonus, positive Babinski. She had toe walking and a spastic gait with a GMFCS of 3. She had some clumsiness, poor fine motor coordination, and also weak voice and dysarthria. Some workup had already been done. We had a normal brain and spine MRI. There were no abnormal findings in the ophthalmology consults. And she is right now treated with baclofen for her spasticity. Next slide. So I think you might have to hit play on the video to start. Perfect. Let's see. So this is her at the age of 1 year, and we see that she is indeed toe walking. She has some slight scissoring, and she needs a little bit of support for her unstable gait. At the age of 2 months, we see that she the disease is a little bit progressing. She still has toe walking. She has the slight scissoring and the instability in her gait. See this more right now when she's rocking, and we see her from behind. This is her at the age of 3 years. She is wearing AFOs, and she needs to hold on to the handrails and needs quite some support by her upper extremities to be able to walk the stairs. This is at the age of 4 years. We see that the spasticity is progressing. She is wearing the AFOs. The scissoring is becoming more prominent. And this video demonstrates her troubles with fine motor coordination. And I think we can skip that one for time reasons. A little bit shorter. Okay, perfect. So this is her family tree. We see that there is no significant family history. She's the patient in red. Next slide. And when we were looking for an autosomal recessive mode of inheritance, we found these 2 compound heterozygous variants. The first variant is a missense variant inherited by the mom with a low MAF, damaging computational prediction scores, and previous research have reported this variant as likely pathogenic. The other variant is paternally inherited. It's in the splice region, also with a low MAF. The prediction scores showed a more possible benign effect of the variant, and it was previously reported as a variant of uncertain significance. Next slide. We then looked for phenotypical associations, and we found 2 reports on OMIM, a big database showing that there are 2 syndromes reported with this gene, both autosomal recessive. One is primary coenzyme Q10 deficiency type 7, one is spastic ataxia type 7. Both conditions have a variable penetrance of symptoms, variable age of onset, and we saw a certain phenotypic overlap. On PubMed, there were some reports of neonatal encephalomyopathy, cardiomyopathy, and respiratory distress syndrome. Next slide. Then we looked a little bit more in detail, and we saw that there were 2 recent reports from the beginning of this year for biallelic variants in COQ4 causing indeed hereditary spastic paraplegia. The patients described in this cohort were a little bit different than our patient. They had more complex symptoms. They were also a little bit older at the age of onset, and this led us to writing a case report about this. You can see all the variants that have been reported on this gene in the graph. The variants in red are all variants reported for spasticity, in patients with spasticity, the ones with that are underlined with ataxia, and the ones with the green square around. These are the variants that we found in our patient. And this shows the broad phenotypic spectrum in this disease gene. Next slide. And why is this finding so important? So one is it has direct therapeutic implications. The papers have found that there were fibroblast studies in patients with biallelic COQ4, mutations in COQ4, and they showed that the patients have a deficiency in COQ10, so we started supplementation of this of COQ10. And then we also started screening our patient for cardiomyopathy as a matter of prevention. Next slide. And this brings us to our second case. It's also a girl, 3 years old, with a normal birth and neonatal history. She has progressive spasticity in the lower limbs, which started showing at the age of 18 months. She now has hyperreflexia in the lower limbs, clonus, positive Babinski sign, some toe walking, spastic gait, ataxia, delay in motor and speech. The workup in the brain and spine MRI was completely normal, as well as the neurometabolic screening. Prior genetic testing was negative, and the girl gets baclofen for her spasticity. Next slide. This is the family tree. We see some history of depression in her family on the paternal side, on the maternal side, some intellectual disability, but no one in the family showed similar symptoms like our patient did. Next slide. This is her at the age of 14 months, and we see that she is walking quite normally for a girl in her age. We don't see any evident signs of spasticity. And this changes now just a couple of months later. She's 21 months old. We see the spasticity, we see the toe walking, we see the unstable gait and a broad-based gait pattern. This is 3 months later. And we do see that the disease progresses when she starts walking. Pedro, maybe you can move a little bit forward in the video. Yes. She is experiencing more frequent falls throughout the year. But overall, at this age, her symptoms are more stable with the spasticity we can clearly see right now. Okay, next slide. And when we assessed her genome for autosomal recessive variants, we found those 2 compound heterozygous variants in WNT1, one nonsense maternal mutation with a low MAF predicted to undergo non-mediated decay, one paternal variant in the splice site region, also low MAF, with predicted impact on the splicing. Next slide. And looking at phenotypic associations, there were some associations with infantile onset recurrent acute liver failure and skeletal abnormalities. Then there was also a recent paper that described 3 patients from 2 families that have indeed early onset progressive spastic paraplegia and ataxia, but also additional symptoms like dysmorphic features and optic nerve hypoplasia. And interestingly, the variants that were reported, one variant is the one that was inherited by the mom in our patient, and the second one is really close to the variant that was inherited by the father. Next slide. And we then decided to take a closer look at the splice variant to better understand the function of this variant. And what we did is we started at the DNA level, the 2 up in the left corner, and this variant was predicted to abolish the splice site. We then moved over to the right part where we performed RNA sequencing, and this showed that it didn't affect the splicing, but effectively led to a deletion of the last amino acid of the exome. And to confirm our suspicions, we performed some lipidomic study. I'm just going to talk briefly about it, which showed that our patient behaved just like the patient that have been previously reported. Next slide. And also this diagnosis was really important for our patient because it now allows us to screen for his liver functions, especially in times of disease, and of monitor him for ophthalmologic abnormalities. One other important thing to keep in mind, the patients that I show you here, they were really young, so also the questions of family planning are really important for the families. Next slide. To talk about the interim results of our study. Right now, of the patients that we have analyzed, 49 patients, we could find a diagnosis for 20 patients, could identify candidate gene for 17 patients, but for 12 patients, we could unfortunately not identify any variants of interest. Next slide. To talk a little bit about the additional diagnostic yield, so we could identify a diagnosis in 40% of our patients. And if we look now in candidate genes and in solved genes, most of those genes and variants were indeed identified on the exome level and not on the genome level. So the additional diagnostic yield of whole genome sequencing in our cohort is 15%. Next slide. To talk about the outlook of our project, so we are part of a pilot project for long-read whole genome sequencing with the Broad Institute. You already heard a lot about long-read whole genome sequencing, so I'm going to keep it brief. Inclusion criteria at this time point is that we don't have a genetic diagnosis despite short-read whole genome sequencing. For this specific problem, we also need parental DNA, but we will expand this in the future for all patients, even if parental DNA is not available. And the aim of this project is to determine the diagnostic yield of long-read whole genome sequencing in rare diseases. Next slide. So long-read whole genome sequencing, we get reads from 10 to 100 kilobase pairs up to 1 million. There are two big different technologies, PacBio and UNT. I'm just going to keep this brief. Different strengths and disadvantages of this approach. So one is we can detect repeat expansions and complex structural variants. And we can also do something that is called de novo assembly. So the first step when we make all these puzzle pieces of the DNA, the fragments, we have to reassemble them. And we often do it versus a reference. And this reference can have systemic errors. And long-read whole genome sequencing allows us to do a de novo assembly. Disadvantages of this approach right now are relatively high costs and also higher error rates of 10 to 15%. Next slide. And why is it so important to actually know your diagnosis? So we always want to go from disease to know what to target in a therapeutic item. And for this, we need to understand what's the mechanism of disease. This is something that Darius had also talked about. So there are different disease mechanisms in the genome, molecular mechanisms that allow us for different techniques in gene therapy or amendable therapies. Next slide, please. And apart from optimizing treatment and new treatment options, there are other advantages of knowing your diagnosis. Next slide. This allows us to start with, to be an entry point for research studies. We can, next slide. We can also help with family planning, next slide, help with counseling, better genetic counseling, and the two cases that I brought you here really show how important it is to know your diagnosis for monitoring and early intervention of possible consequences of your diagnosis that might not display right now. Next slide. And it also helps to build communities, which are really the foundation for a lot of research that is happening right now. Next slide. So to summarize my talk, the preliminary results are that we can see an increase of the diagnostic yield in 40.8% in whole genome sequencing, which is mainly attributable to exome-level reanalysis. We have a 15% additional diagnostic yield of whole genome sequencing. Genetic diagnosis can yield actionable clinical findings, and additional sequencing and analysis methods are warranted. Next slide. So to close my talk, I would like to thank all the patients and their families that are really the foundation for and driver for all our research, and also the big team that you can see on the pictures there, especially Dr. Darius and Amy Tam, who are also here in Tampa with me. Next slide. And I would also like to mention that we have the two studies where we are still recruiting patients. If you have any interest, don't hesitate to reach out, and that we have the Movement Disorders Program at Boston Children's. Thank you very much.
Great job. Hey guys, if you want to, because we need to move on to get to our next segments of stuff we have to do, remember, her email is in your program. If you want to email her directly, she'll give you all of the answers. And
I'd be happy to.
yes, she will, because she's so great. And also, remember, all of these presentations are being recorded, and they're going to be on our YouTube channel in a couple of weeks once we put them all together and be uploaded through the website, okay? All these will be out there, so we'll get to look at them, and you can go at them at your own speed. You can speed them up and slow them down and look at all the quality research, and you can blow up the slides real big in your home, because we know you all have 90-inch TVs in your living rooms, right? So you can put it on your big 90-inch and see all the graphs and the data up real close. Okay, so remember that, couple of weeks, all of these will be out on the SPF YouTube channels. Okay, got it? We're going to take a quick couple of seconds here and get ready for some more stretching. Here we go.
I hope not too many people are leaving us.
There he is. All righty, Zachary, here we go again. All right, you're on.
Yeah, I have this. Okay, so just give me 1 second. For everybody who can, let's stand up. If not, totally cool to sit down. And I'd like for you guys to grab at the back of your chair if you are one who's standing up with us. What? Too far?
There we go. Sorry, that's on bounce on. There we go. A little bit better.
Perfect.
There you go.
Okay. All right, guys, we're in. So if you need the balance, feel free to put one or both hands on your chair. And whichever, or the length which is most comfortable, we're just going to do some small kicks with our left foot. Now, if you can bring your leg up, totally cool. If not, tiny wiggles. And it really doesn't matter which leg you start with, just getting some leg swings in. I hope you're here. I spoke to somebody, gave me a recommendation for today. Thank you. You're welcome. And let's switch. I was hoping you were here. Good. Okay, how many of us have tried a squat before? Okay, so let's see. So we're going to grab a chair if we're comfortable. If not, classic, we'll just squat down here. Now, if you are not new, or if you are new to squatting, let's take it slow. So I don't want you to try to touch the ground with your butt. Let's make slow, it might look or feel weird. Slow squats until you find a depth that's good. And totally grab the chair if you'd like. If not, just keep it here. Something I said last time that I just want to push into everybody's brains again is simple concept that positive progress is possible, even if there are no cures. There are things that we can do every day to make sure that we're safe. So let's stop squatting. We're going to stand up. And now we're going to move our hips a little bit. So you could start with your left leg, and we're just going to swing it all the way out. Now, same as our squats, maybe you might be a little uncomfortable going all the way up, so you could try moving your leg up a little bit, and even if that's too hard, you can do little circles right in front of you. But if you are holding onto a chair and you have good balance, I want you guys to try to lift that leg up. You should feel it in your hips. It might hurt a little bit, be a little tense, but it's supposed to. And let's switch. Okay, and one more time through. we're just going to do some kicks like we did in the beginning. And let's switch sides. Okay. Now everybody sitting or standing, take a deep breath in, bring our arms all the way up to the sky. Just going to exhale, bringing our arms back down. Let's do one big inhale, bringing our arms all the way up to the sky. And like we did this morning, let's just release and hold, if you can. If not, you can totally just stay up and keep doing what we had before. Let's take one more inhale, bringing our body all the way up. And let's reset. Nice work, everybody. Nice work. So let's bring our arms straight out in front. Might be a dance. I'm not really sure. Forgot the name of this, so we'll pretend it's a dance. Get our arms moving a little bit. And let's switch. Let's pump our arms up in the air. I don't know about you guys, but I'm already feeling this. My shoulders are starting to hurt. And release. How does that feel? Does that feel nice so far? Okay. So I want you guys to follow this movement. We're going to bring our arms up here and then go straight up in the air. When you get up here, I want you guys to try to extend those shoulders up as high as you can. When you're bringing down, release your shoulders. Okay, so for this one, to start off, I'd like for people to grab the chair, and if it turns out being a little easier, then let's go without the chair. So we're gonna work on our hips a little bit. Let me come out to the front. I have HSP, so I'll hopefully have my balance while I show this to you guys. So this is another exercise to open up our hips. So we're going to start, any hip. We're gonna bring our leg up, and we're rotate it to the outside. We did it this afternoon, where we were rotating our feet in and out, except this time we're actually going to do the motion. So we bring our leg up, try to turn it to like a 90-degree angle with the bottom of our foot. And if you do feel like you have balance, totally try it without the chair. I just wanted to make sure everybody was good. And we can switch sides. You guys feel that one in your hips? Yeah. Good, because I do as well. Okay. So once again, we can do the sitting or standing. We did this afternoon as well. We're going to bring one foot forward. So I'm starting with my left. You can choose whichever. Going to have our right foot back. Our right foot is going to stay, or our right leg is going to stay a little bent. And we're going to keep our left leg as straight as we can. And from there, we're just going to slowly try to reach down for our foot. Grab a chair if you need, please. When I was up here last time, I could not stop laughing in my head because my parents used to bug me about stretching all the time when I was a kid, and I never stretched until I got older and I realized that I had to. So I find it very funny that out of everybody, I'm the person talking to you guys about stretching, considering my parents bugged me every day of my life to stretch. And let's just switch sides. We got one more. And just like that, thank you everybody for our stretches. It is 3:45. We have about 15 minutes until our small groups.
Way to go. Thanks, Zachary. You're the best.
Thank you.
Okay, everybody, you know where you got to go. You've got your...
Thank you for all the wonderful participants.
You know where your breakouts are and where you're going to go. PLSers are down in this hallway again. The child onset in the front of the building on the second floor. Okay, and we got podcaster Kim Sanchez in here in just a few minutes. We'll see you soon. And Zachary, by the way, is also in the front of the building on the second floor for his breakout session. So thanks, Zach. You're the best.
Well, good afternoon, and we're getting toward the end of this second day with a lot of good information. You're hearing from speakers, and we know some of it may be a little more difficult to see, and we can't ask questions of some of the folks who are getting with us by Zoom, but there are speakers with research that we felt like, Jim, we needed to hear from and be updated on, and so it's good to hear from them, Dr. Zeugner and Dr. Calamé, and as we do this virtual each year, we're learning a little bit more about how we might do things differently, and we're going to try to find a way, working with our team, that maybe we can figure out a way next year to have you all be able to call in as we do that. That would be our goal. We'll be working in that direction and see what the possibilities are, but we're glad that you've joined us, glad that you've been able to get this information with all those things going on research-wise. With me now is, you guys all know, our fundraising chair and past president and whatever her title goes, with Jim Sheehan. And I've asked Jim, we've talked a lot, obviously, about all the different functions of the foundation, Jim, awareness and outreach, and the fact that 60% of our people are first-time attendees, and all those things are important, but we know at the very top of our list of priorities is to raise money to move this research to a point that hopefully you and I one of these days have a treatment. So I wanted to get you on to talk with us a little bit and share where we are. It's almost halfway through the calendar year. How are we doing fundraising? What are we looking forward to the rest of the year? So let me just hand it to you to talk to us about that.
Great, thank you. And the good news is we're doing very well. We are right at halfway through the year. To date, we have raised just over $525,000, which is significant. That's actually about 5% up higher from last year at this time, so that's good. We've got some events coming up in the near future. I'll go over those in just a second, but in your spare time, if you can, go to the webpage and look at all the different types of events that we do throughout the year. Look at something that works for you, pick it, and make it happen, because we need everyone out there to participate, make personal donations, reach out to family and friends, share your story, make sure everyone knows the difficulties you deal with, and ask them to contribute, because this is going to hopefully bring treatment options and cures in the near future. What we have left this year so far with fundraising is we're actually kicked off the 5K run, walk, and roll. It's available now through the end of October. You can go to the website and look under events, and you'll see the list of events. Go to the 5K page. It'll list all the information on how it works, and it'll allow you to take a link to the peer-to-peer fundraising page. It takes just a couple minutes to upload a picture, set up your own personal page, and then you can share that with all your family and friends. You can do that through Facebook, emails, whatever you want to do. We're hoping to raise $80,000 this year, and again, this will go through the end of October. Shirts this year is what I have on, this dark blue shirt. That's actually for the folks, anyone that goes on and sets up a peer-to-peer page and raises $100 or more will receive this shirt complimentary. So look forward to that if you want. You don't have to be a superstar athlete to participate. Find your activity. The main purpose of this is to become physically active or be physically active and to raise money for the foundation. So swim, golf, canoe, tennis, golf, run, walk, roll, whatever it is you can do. Find something to be active for an hour or so. If an hour is too long, do something for 15 minutes, 3 or 4 times a week, or take 2 weeks, whatever. Make it your event, but be safe about that. After that, we'll be sending out the annual report. Greg and Norma and some other folks will be working on that. That's really just to show our community members how well we've done in the previous year. So this will be results from 2023. That will be available here in the next month or so. And that's a great opportunity. A lot of people get excited about what we announced, about how much we raised, and about research. So a lot of times folks give through that way. We'll be wrapping up the year with our year-end giving, including Giving Tuesday. So the first part of November, you'll be getting a letter in the mail, emails and stuff like that to promote our year-end. Usually we raise a half or even better than half of our annual money through the year-end campaign. So if you haven't given yet, please do if you can. Please reach out to your family and friends. And if now is not a great opportunity, please do so this year if you can. And we really need to step up and try to raise as much money as we can, because again, I think we've got some treatment options and maybe some cures in the near future. Lots of positive information here at the conference. We had at least 8 speakers that talked about positive results. Gene therapy, one researcher has a list of medications he's going to start doing patient trials on. It's just exciting, and they're working together. So it's a great opportunity to get involved and make sure you support the foundation, which will hopefully bring some good answers for us in the near future.
Jim, as soon as we get home from this event, one of the things on our top priority at our home is our annual Golfing for Rare Disease event that we do the Saturday after Labor Day every year. A number of golfers, sponsors will participate in that, and a half a day of golf, we normally are able to raise about $20,000 to $25,000. And if there are other golfing families out there, folks who know how to make those things happen, we've got a very simple model we'd be glad to share with you that maybe can turn your love for a sport into some money for the foundation where you're located. If you'll get in touch with us by email or in whatever way is easiest for you, we'll be glad to share those ideas about how to do that. And it does take some work prior to the event. I mean, you don't just wake up and go do the event. There's a lot of preparation, but it's worth the time and worth the effort, because $20,000 to $25,000 for an event's not a bad day. Right. So we'd invite you to do that. Jim, as we go through this process, you and I, you as fundraising chair, and Norman and I often get questions about, well, what's happened with the money we've donated in the past? How is research going? What do we know? What can you tell us? And I tell folks, we're not scientists and we're not researchers. It's hard to answer that question. But both to Dr. O and Dr. Fakhari today clearly said all the research that we and other foundations and contributors have funded in the past have brought the research arena to the point now that we really are at the proximity, we hope, of finding some things that can make a difference with a treatment. The CERN that we established last year, creating the 10 centers at this point, that clinical trials might be easier and quicker to do with the way they're putting that together. So a lot of good things happening. We assure you that a treatment and a cure is not happening fast enough for either of us either, but that's why we keep working as hard as we are at this to try to get that to happen soon. We have family members, we have friends, and we want to see their lives made better in the future as we work to do this. So that's what's going on this year with fundraising. We ask you to continue to doing what you have done well in the past, and let's have the best year this year. And I'm confident, Jim, I really believe, based on how things are going now, that come December 31, January 15, we're going to be able to say 2024 was the best year ever, and that we can get more research done than we've ever been able to get done before.
$1.5 million. Let's do it.
Thank you for your leadership with our fundraising committee and our fundraising effort.
Thank you.
Let me share with you guys, this will be the last time I'll talk with you today. In just a few moments, Kim Sanchez is going to come and share with you and folks here who want to learn about how to use social media, how to talk to folks. You may think, I can't do that, I'm too shy, I don't have the personality. Well, I want to tell you, if you look at social media very long, you'll find that you've got as pretty a smile, you've got as much personality as a lot of the folks that are out there doing that. It just takes some confidence and some willingness to do that. And that's why we have Kim here. She has HSP, as she shared with you yesterday. She wants to help us become better at getting our word out through different social media platforms and in other ways. So we hope you'll hang on and listen to her, and that you might think about doing that somewhere you are, so that your friends and family will know more about your condition and about the opportunity to raise resources to help us find these treatments and cures. We'll be back in the morning tomorrow. Two things happen tomorrow. We'll hear from all of our committee chairs about the work of their committees, and we always invite you to pick one of those committees that you might have a talent or gift or experience in, because we can use everybody's time and everybody's work as we work on these agendas and these items of work. Then following that is what's always kind of the highlight for a lot of people. Dr. Fink will be the last doctor who will speak, and he'll be answering questions, and that will be in the morning as well. So we hope you plan tomorrow to be with us and participate in watching that as well. In the morning, I'll come on at about 8:25 just to kick the day off and tell you again how things will go tomorrow. But I won't be seeing you anymore today, but hang on for a few minutes, listen to Kim, and let's get this day done. You guys enjoy that presentation, enjoy this evening, and we'll look forward to seeing you at 8:25 in the morning. Have a great evening.